in the Koch Institute building. I'm in the Department of Biological Engineering. And I just have to say, based on that talk, this is going to be an amazing meeting. There's not a single project in my lab that doesn't benefit in some way from Carolyn's work. So thank you to Carolyn. I'm going to introduce a session with a number of exciting industry speakers, but before we do that, I just have a couple of housekeeping items. We do have folks who really want to get into the auditorium, so if you let them kind of get into seats, that would be great. I don't know about you, but it's starting to get a little cold after that 102-degree couple of days. So I decided to put on my new Koch Institute swag for the 50th anniversary. If you are a Koch Institute member, please pick up your swag and show some team spirit. And then Jeff also wanted me to say that there's a coffee break following Peter's talk, and then session 1 will resume at 11 with Gerald's talk. So with that, I have the pleasure of introducing Peter Center. He joined Seagen in 1998, where he built and led the chemistry group that carries out research in cancer drug design, antibody drug conjugate technologies for cancer therapy, and protein chemistry. Several of the molecules generated in Peter's lab have entered clinical trials, and 4 have been approved, and he'll talk about those today. Dr. Center has authored more than 160 scientific publications and holds many patents. He received his bachelor's degree in biochemistry from the University of California, Berkeley, and a PhD in chemistry from the University of Illinois. He did his postdoctoral research at the Max Planck Institute of Experimental Medicine in Göttingen, Germany. He serves as senior director on molecular cancer therapeutics and is an affiliate professor of bioengineering at the University of Washington. And just a side note, one of the students that I mentored as a graduate student in the chemistry department at Harvard, who's a close friend today, Chris Newman, has been mentored by Peter Center. So I've heard a lot about your amazing abilities as a mentor over the years. So with that, his talk title is Antibody Drug Conjugates for Cancer Therapy, from early stage research to clinically approved drugs. Let's welcome Peter.
Is the microphone on? Yes, it is. Oh, thank you. I can't believe that I'm following Carolyn Bertozzi giving a talk. It's not the easiest thing to do, but I've been a huge fan of Carolyn Bertozzi for many years. And so, as in the introduction, I came from Berkeley. I went to University of California, Berkeley, and that's where I got to know Carolyn. And then when she went to the dark side, and moved over to Stanford, I invited her, me and David Rabuka on the left, invited her to a Stanford-Berkeley football game. And it's a big game, that's what they call it. And the funny thing about it is that I asked Carolyn, who are you for? You know, you just spent 10 or so years at Berkeley and you've just been at Stanford for like a few months. And she said, well, I've never been to a football game before. It was kind of funny. And anyway, one of Carolyn's big claim to fame, and she didn't mention it in her presentation, is she got me to wear a Stanford uniform. And I only did it because it was so cold. But anyway, these are some historically significant antibody-drug conjugates that were developed by pharmaceutical companies in the early days, when it seemed conceptually simple to take a drug and put it onto. We got it back. Is it? It was. Sorry about that. Anyway, it seemed conceptually simple to just take a drug and put it on an antibody, but it turned out to be more complicated than we thought. And so, some of these early examples really began to highlight the challenges. Eventually, Mylotarg on the right did get approved, but it turned out to get unapproved, withdrawn from the market for many years because it was so toxic and difficult to administer. And during all that time, a lot of lessons were learned, and they had to do with how maybe some of the rules could be abided by in order to make conjugates more effective and safer than they were before. And they included controlling the conjugation, having potent drugs, controlling the pharmacokinetics, and a number of other factors. And these were all things that we took into account in the early days of Seattle Genetics when we got into the program and decided to make antibody drug conjugates. And so I'm just going to very briefly go over some of the things that we did. At the beginning, we started to work with a professor at Arizona State University named Bob Pettit, and he had isolated really potent drugs or potent molecules from the Indian Ocean sea here. One of them was called dolastatin 10. We were really interested in dolastatin 10 because it was much more potent than other antitubulin drugs that were currently used in the clinic for treating cancer, and that was the mechanism of the drug. And it was also a molecule that we felt that we could synthesize and modify in order to optimize it for drug delivery. And that's exactly what we did. We made an analog of dolastatin 10 called monomethyl auristatin E, shown in the bottom left. And when we took that drug, put it onto an antibody with a protease cleavable linker, somewhat similar to the linker that Carolyn described for the Daiichi Sankyo molecule with the GGFG. But this is a dipeptide valine citrulline with a self-immolative spacer in between. We could see that when it was delivered inside cells, that there was a dramatic effect on the polymerization state of tubulin, which is shown in red on the bottom left-hand portion of the slide, where untreated cells or cells that had just briefly been treated with the conjugates had polymerized tubulin, but then after a little while, the tubulin became depolymerized and compact, and the cells were unable to undergo division. And we took this technology and tried it out on many different antibody platforms, many different types of diseases. And we did something that biotech companies at the time, or even pharmaceutical companies at the time, had never really considered doing, and that was we decided to apply it to a disease called Hodgkin lymphoma, which was kind of an orphan disease. It's not very common, but it affects younger people, and around 25% of the people who get it, and these are people in their teens typically, don't respond to chemotherapy and have a terrible time with the after effects of the aggressive therapies that they receive. And so we thought that there was a real unmet medical need here, and that was what we went after. So we had an antibody against CD30, which bound to the malignant drivers within the tumor, and this is histology showing the brown cells are the cells that we're targeting, and that was really what we decided to go after clinically. And so here's a slide that you don't often see. It's 16 years of clinical work from phase 1, phase 2, and phase 3 of the drug. So the phase 1 clinical data was kind of astonishing because when you're in the drug development business, the last thing you expect is to see responses in a phase 1, but that's exactly what we saw. It was really kind of amazing. And this is a patient who, on the left, had around 5 previous chemotherapeutic treatments and had progressed through all of them, went on the trial, and the dark areas are the tumor except for the bladder and the heart, which is also dark. And then after just 2 treatments, that patient had a complete response. And it was data like that in phase 1 that made us think that this could be a drug. It might really be working. And so we did a phase 2, and that's shown in the bar graphs in the middle here with each of the patients in the clinical trial. And we saw that the vast majority of patients responded to therapy. And these were 2 different diseases we were treating. In the upper part, we were treating Hodgkin lymphoma. In the lower part, it was a form of non-Hodgkin lymphoma. And so it was really effective. And based on those data, the FDA approved the drug, as Carolyn pointed out. And then, but it was required for us to do follow-on clinical trials where we were actually comparing it to standard of care. And so we took our drug, at this time called Adcetris, combined it with chemo, some of the chemotherapeutic regimen that patients got when they had Hodgkin lymphoma. This is frontline patients now that hadn't been pretreated, and we saw that the effects were better than the full course of chemotherapy. And importantly, there were fewer side effects, and the risk of death went down by about 50%. And so based on that, the drug got approved for treating frontline Hodgkin lymphoma. And we took the same type of drug linker platform that I described to you, which was the dipeptide with monomethyl auristatin E, and applied it to different antibodies. And this is an antibody against bladder cancer or metastatic urothelial cancers. And it's a difficult disease to treat because patients who get bladder cancer tend to be older and they don't really withstand chemotherapeutic treatments. And so we developed this drug for treating bladder cancer and combined it with a drug that takes the brakes off the immune system called pembrolizumab. And there were a number of reasons why we chose that specific combination, because we thought that mechanistically there was an aspect of immunological regulation that really participated in the activity of auristatin drugs. And so taking the brakes off the immune system seemed like a good combination. And we saw that, in fact, the effects were really pronounced. And this drug also got approved, and it's now approved for treating frontline bladder cancer. And these are survival curves where you can see the difference between the combination of these two targeted agents. It's not really chemotherapy, is much better than just chemotherapy alone with fewer side effects. So it was an important aspect. And as Carolyn pointed out, the technology that we were using in developing these drugs was not perfect. It had some serious flaws in that we knew that it was heterogeneous because we had to settle for heterogeneity because we could only put on 4 drugs per antibody on the average and maintain the pharmacokinetic parameters of the antibody. If we put on 8, there was too rapid elimination from the body. But then another thing that we found during the course of our clinical work was that the drug was coming off the antibody over a period of time through a retromical reaction, a fragmentation reaction. And in the field, it was always believed that maleimides, when they reacted with cysteines, were really stable, but we found that, in fact, over time, they do fall apart. So we knew that that was going on as well. And what we did then was we went back to the lab and redesigned what we considered to be a perfect conjugate, in that it was homogeneous. We could put on 8 drugs per antibody because on the right, you can see it's much more hydrophilic. It has a small ethylene glycol linker. It has a glucuronide instead of a peptide for cleavage. And it's the same drug. And we also designed a maleimide that didn't undergo the retromical fragmentation. And so we thought that this was going to be the answer to everything. And it wasn't. It was really interesting because what we found was that when we administered this so-called perfected drug, it completely depleted the bone marrow in animals that received it. And I should point out that CD30 is present in the bone marrow in cynos. And so by really efficiently delivering the drug to CD30 positive cells, we actually eliminated the bone marrow and found that the drug was not therapeutically useful. And so there is some value in the imperfection that I told you about, that having the drug come off over time turned out to be a potential advantage, at least for the format that I described. And we weren't the only ones who saw that. Genentech had been playing around with some very stable drug linker formats and showed when they compared it to the unstable forms that they began to reveal ocular toxicity that wasn't present in the unstable format. So it's, there's still a lot of development to do in this field to try to pick apart how we can control the activity and the toxicology. And it's really important. These are 2 phase 1 clinical trials where we look at the objective responses in patients that receive these conjugates at increasing doses. And these are the more classic, unstable, heterogeneous forms of conjugates that I described. And what we found is that in these 2 different studies, the efficacy goes up as we give higher doses, but we're running up against a maximum tolerated dose. So the goal of much of the work in the field is to try to circumvent this, to make conjugates that are more safe and that can be given at higher doses and achieve higher levels of efficacy. And so the way that people are doing this is they're beginning to look at every part of the conjugate. Like one of the things, of course, is how you deliver the drug. Is an antibody really the best way to deliver a drug? Can we do better with something else? Or can we modify an antibody to make it better than it classically is? The linker is really important. Can we make linkers that would be, let's say, more selectively activated in the tumor compared to a normal tissue? And then the warhead. I talk about MMAE, but there are other drugs like the camptothecans that have turned out to be amazingly important. Are there other drugs beyond that that would provide activity and safety as ADCs? And I'll talk about that a little bit. So I'll start with the antibody. One of the issues with antibodies is they can deliver molecules to cells, but a lot of times they just sit on the surface. And as Carolyn pointed out, sometimes that's okay, because if the tumor is releasing proteases, or if there are proteases in the tumor microenvironment that can cleave the drug off, then the drug can still come off. But if we're trying to get these things into cells, it can be a limitation if the antibody just sits on the cell surface. And that's seen with Herceptin on the right, and also with our clinically approved drug, where the vast majority is not going inside the cell, it's sitting on the outside. So then the question is, what can we do about that? In some cases, we may want to try to improve internalization, and so I just thought I would provide an example. This is an example from Genmab, where they made a bispecific antibody, so it's HER2, and the other arm of the antibody is a receptor called CD63. And what they showed was that when they had this bispecific antibody that bound both HER2 and CD63, it went inside cells much more efficiently than just HER2. And in fact, it depleted HER2 from the cell surface, as shown here. And the effects of the combination of this bispecific were better than just the HER2 conjugate. And it illustrates that there may be a way of improving ADC activity if we can improve the way that it goes inside the cell. And there are many ways of using molecules on the outside of a cell. This is one of the things that Lycia is doing. It's one of Carolyn's companies, to improve delivery within the cell. And speaking of antibodies, there's been a lot of interest in non-antibody scaffolds. And I can tell you that therapeutically, non-antibody delivery systems are beginning to have a gigantic impact in clinical oncology, like with the advent of drugs like Pluvicto, which is a small molecule that targets PSMA, and it has a radionuclide attached to it. It's a really important drug, and there's a whole bunch of these things now that bind to hormone receptors that look really interesting. They bind to somatostatin and other things that are non-antibody scaffolds for the delivery of cytotoxic agents. And there's been a huge interest in other motifs besides antibodies. So what I thought I would do is take you through some of the lessons that were learned with a molecule that went pretty far into clinical development. And this is called the bicycle therapeutic. This is work that was done by a company both here in Boston and in the UK called Bicycle Therapeutics. And what they had done was make a small peptide through a phage display library that could bind to tumor-associated antigens. In this case, it bound to an antigen called Nectin-4, which is the same target that the antibody that I showed you for bladder cancer binds to. So it binds to Nectin-4. It has the same drug attached to it, MMAE, and it really works. In their preclinical studies, they could treat small tumors and the tumors disappeared, or they could wait for the tumors to get really big and treat it with this bicycle therapeutic, and the tumors disappeared. It looked incredibly promising, and it's only 4,000 molecular weight, so it's about a 40th the size of an antibody. And so given that it's a very small molecule, it can extravasate really quickly, get out of the blood supply, and clear from the body quickly, it's not too surprising that after about 3 hours of administration in a patient phase 1 clinical trial, it drops to an undetectable level. But the thing that wasn't expected in this clinical trial is this inset, which you may not be able to see so well. And that is the active drug concentration, MMAE, in the blood. And what you see here are days, this is not hours, where you have high levels of MMAE circulating for days. So I think that this construct wasn't perfect by any means, because even though it was clearing quickly, it was going probably into the kidneys or to peripheral tissues, out of the blood, and eventually just releasing the cytotoxic payload. And what was really interesting about it was the maximum tolerated dose of the bicycle therapeutic compared to the approved drug, enfortumab vedotin. And what they found was that you could give 5 mg per meter squared, which represents 18 micrograms per kilogram of MMAE, which is exactly the maximum tolerated dose of the full conjugate. So I think there's hope and promise for small molecule delivery agents, but this one was probably not designed appropriately, and I think the field needs a significant amount of work to get to a point where it really looks more encouraging. One of the other things that people are looking at are the peptides. There's a tripeptide that is cleaved by enzymes other than cathepsins that is a lot less toxic in terms of neutropenia or depletion of reticulocytes or in the bone marrow, and it's in a clinical trial now. In preclinical trials, it has an MTD that's about twice the MTD of the dipeptide, and we're hoping that something like that could constitute a significant improvement in therapeutic efficacy and tolerability. Lastly, as far as linkers go, this is an interesting study where we took the glucuronide linker and looked at how much drug is released inside the cell compared to a dipeptide linker, and saw in the blue there that we're getting like 2 or 3 times as much drug released inside the cell. And we think it has something to do with the fact that the drug is being released in early endosomes in a very early stage in the endocytic pathway, as opposed to the dipeptide, which has to get into a lysosome. And so that's in a phase 1 clinical trial as well. So there's a lot of opportunity space in the ADC field with new linkers, new targeting motifs, and even new drugs. And I think that the field is really in the early stages right now. I don't really have time to go through a lot of the work that's been done, but this is an interesting drug that was developed by Immunomedics with the camptothecin. It defies all of the principles that I told you about stability and everything PK, but it works. It really does. But the question is why does it work? It may not work because of the mechanism that we think. It may just be releasing high concentrations of active drug, SN38, in the circulation. And in fact, the amount of SN38 in circulation of patients that get Trodelvy is much higher than the active concentration of SN38 in patients that get a prodrug of SN38 that's approved. So there's still some questions as to how it's working. And Carolyn had already pointed this out. There's some questions about how some of our targeted ADCs work when we see data where Herceptin conjugates, TDM1, actually work on tumors in patients that are antigen negative or ultra low in antigen. And so then the question is, what is going on here? And it may very well be studies like this that were done by a group at Duke and just recently published that shine light on it. And that is what they found is that this TDM1 conjugate, which has a peptide, is active in cells that have a lot of HER2, ultra low, or even negative, as opposed to other conjugates that have to go inside the cell in order to be activated. And it suggests that there may be mechanisms that remain to be understood. They're suggesting that it's a release protease that's causing that. And they're not the only ones who have seen this. We recently published our data with brentuximab vedotin in a disease called diffuse large B-cell lymphoma, where we're combining it with chemotherapy. And what you see here is better efficacy in patients, and it led to the approval. But one of the complicating things about the study is that patients with diffuse large B-cell lymphoma are CD30 negative. It's true. They don't have CD30 on the tumor. But what we think might be happening is that in the tumor microenvironment, there's a lot of macrophages that can lead to the release of the drug. And we've seen that in preclinical studies. And it just highlights that ADCs are interesting, impactful, they're approved, but they're complicated. And so there's a list of all the approved ADCs. I'm not making up any of these numbers. There's 3,970 current clinical trials underway. There's 573 companies that are almost fully focused on ADCs in the world. And there's a lot of advancements that have been made in the field. I think that this list here will double in the next 5 years easily because of all the late-stage clinical trials. But I think that once we understand better delivery vehicles and new drugs and bispecific co-conjugate technology, and really understanding the mechanism better, we'll be able to make ADCs that are even better than the ones that are currently approved. So thank you very much. I appreciate the opportunity to speak.
Thank you, Peter. I have one question. First, I echo your notes about benefit of imperfection. But the question is about the neutropenia observed for the peptide drug conjugate compared to the Nectin-4 ADC. Looks like the neutropenia is worse for the peptide drug conjugate.
I'm sorry, did you ask if it's immunogenic?
Yes, sir. So,
oh, no, we haven't seen that. I mean, we see a lot of other problems, but that's not one of them. So we've looked, yeah.
Yeah, neutropenia looks like is always some issue with ADCs, but looks like for the peptide drug conjugate, it looks like even worse. What's your thought on that?
I'm sorry, I didn't understand the question.
So in your bicycle
Yeah.
therapeutics, right, comparison of ADC
Yeah.
and PDC,
Yeah.
the neutropenia is a lot worse for the
Yeah,
PDC, right?
It's such an interesting story, everything about it. But I think that the bicycle technology might have worked if it had been redesigned, or if it had been designed differently. But what they did was I didn't go into it, is they attached the drug to this constrained peptide with a very long disordered peptide in between. And I think that it exposed the drug to have the release of the agent all over the body. And so I think bicycle therapeutics could work really well, but I think that they would have to be designed differently.
Okay. So any thought, in terms of animal models to predict the neutropenia?
I think that animal models could predict, could be used to predict the activities of some of these newer agents better than the ones that, to show that they're better than the ones that were out there before. I think so. Okay. I mean, the bicycle therapeutic is a really interesting example. It doesn't internalize very well. The drug seems to be coming off pretty easily. And, but I think that there are small scaffolds that are currently being explored that look a lot more promising than that.
Okay, thank you.
Mm-hmm.
Hi, thanks for the talk. I'm struggling to understand the efficacy of the HER2 targeting ADCs in cancers that lack HER2. So is it the current view that it's other cells in the microenvironment that express HER2? Is it that you simply concentrate the antibody regardless? Is it that there's a little bit of HER2 on the cancer cells that are called HER2 negative? What's the current view?
Well, this really applies to the breast cancer studies, and there's been other studies where there was a correlation between HER2 expression and activity. And I should say, even in breast cancer, the very high HER2 expressing forms had higher response rates than the ultra low or the negative ones, but the ultra low and negative ones did have response rates. And so I think the idea is that the conjugate is just maybe localizing to some extent within the tumor mass and getting released by enzymes that are secreted.
Yeah, but that would argue that any antibody would do that, and maybe that control's not done, but that's what it would suggest.
I would love to see that experiment done. I would. And I think that it would reveal things that maybe we don't know. I mean, I think it'd be very interesting to have a universal conjugate that doesn't necessarily bind to an antigen on tumors, but just maintains good PK, gets into the tumor mass, and has a very efficient extracellular drug release mechanism.
Right.
I think it'd be interesting.
Thanks.
Hi, Peter. I really appreciated the kind of forward-looking perspectives. So as the field kind of moves towards new payloads or warheads, are there general classes of drugs or, I guess, potency ranges that we're going to need to stay within in order to, as we kind of move away from generally cytotoxic, but stay within an area that still makes sense for ADCs?
Well, there's been a lot of failures with picomolar activity drugs. Some of the really potent DNA alkylating drugs have been incredible safety problems. Yeah. They've just been very difficult to develop, and I think that you need to have fairly decent potency, maybe like nanomolar range potency is something that can be dealt with. But I think another aspect that's really important, I didn't go into it here, is that you need to have sustained target engagement. So that if let's say that you're just going after a kinase that's inside of a cell, with something that can bind to the kinase with nanomolar activity, it's not going to engage the kinase for very long, so it won't be very effective. And I think one of the reasons why camptothecans and auristatins worked as well as they did is the biochemical target inside the cell is there in very high abundance. So I think that's the most critical thing, is not so much the potency of the drug, but how long it can stay on the target and exhibit activity.
Got it. Thank you.
We're going to move on to our break, so let's thank Peter. I'm sure he's willing to take additional questions at the break time.
Sorry about that.